Hello everyone. Let's continue with these mechanisms that regulate body composition and adiposity. And we spoke about the communication between the adipose tissue and the central nervous system. This is the diagram that we discussed on Monday. So, and the idea was that by sending signals to the central nervous system, the adipose tissue can actually communicate what's going on with the amount of energy that is available in the organism, and basically the adipose tissue. So I think one thing that I didn't mention, and I wanna make go over now, and also make the link to the next topics we are going to talk today, is that these changes in the adipose tissue, they don't occur in a short term. What do I mean by this? Well, for your adipose tissue to increase in size or reduce in size significantly to a point that the signals are changed, they don't occur from, let's say, breakfast to lunch or from lunch to dinner. It's not a very quick response. Obviously, your adipose tissue starts to accumulate fat if you are in a positive energy balance, and it starts immediately when the energy is available. But significant changes in your adipose tissue and your fat mass, they don't occur overnight. They take a couple of days, sometimes a little longer than that. Because one day you may eat a little bit more, the other day you may eat a little less. So there is always some kind of adjustment that occurs. So these signals that arise from the adipose tissue, we call, especially leptin, as we mentioned before, we call it a long-term signal. Long-term because if you engage in an exercise and diet program, you want to lose weight, it takes a little time for your adipose tissue to actually shrink in size to reduce significantly leptin, and then to affect your brain. So for it to be sensed and trigger all these other mechanisms that I described on Monday. So leptin, or this communication between adipose tissue and central nervous system to regulate adiposity or fat mass, is what we call long-term mechanism or long-term signal that regulates energy balance and metabolism. So, but what we also have to understand is that even before anything happens in your adipose tissue, there are mechanisms that regulate your food intake and your, let's say, if you're hungry or satiated, that occur in a very short period of time. For instance, when you wake up in the morning that you have not eaten overnight, you feel hungry, or you may feel hungry, and you're willing to eat. So, but as soon as you start eating and you get enough food for breakfast, then the hunger goes away and you become satiated. So there is a signal that has to quickly send information to your brain to say, well, you have eaten enough. The same thing happens from breakfast to lunch. So when you finish breakfast, you're satiated, you're not hungry. But then by the time that noon or close to noon time arrives, you start feeling hungry again. Why is it? Because those short-term signals that were informing your brain to initiate satiety, they disappear or they fade. And then other signals also arise and actually stimulates hunger again. So you have, on a meal-to-meal basis, you have signals that we call short-term, because they control how many times you eat per day and when you're hungry or not on a day-to-day basis. And this adipose tissue mediate or signal mediated by leptin is more of like we call long-term signal, because it requires a significant change in fat mass or in adiposity to either shrink or increase the size of the fat tissue to actually signal to your brain. So with that, I think we need to discuss which are the short-term signals that actually regulate appetite on a meal-to-meal basis. So these short-term signals, they're important for energy homeostasis, obviously, because they either stimulate your food intake or they suppress food intake. So short-term signals They are what we call situational or meal related because they operate to inform specific centers in your brain about the size and timing of individual meals. So when we say specific centers in the brain, most of them are located in the hypothalamus. And they control, again, the size and the frequency, or the size of a meal and how frequent you actually ingest food. So there are two major ones that we know that they operate very clearly on a meal-to-meal basis. Two of them, one is called ghrelin, and the other one is cholecystokinin, or CCK. I think you've heard of them already, cholecystokinin or CCK for sure, because we spoke about it when we were talking about digestion. Because cholecystokinin not only triggers the release of specific hormones, or bile and pancreatic juice, but it also sends a signal via the blood. Also, it communicates with your brain to tell if the food if you have eaten enough or not. So it also plays a role in communicating your central nervous system about the size of the meal that you're taking in. So these two short-term signals, they arise from the gut and regulate food intake on a meal-to-meal basis. So ghrelin and CCK, they have opposite effects. Ghrelin is released during the post-absorptive state or under fasting conditions and promotes hunger. So in other words, if you go to bed, let's say around 10 o'clock at night, and you don't eat anything until the morning after, when you wake up, let's say you wake up at 8 o'clock in the morning, and you have spent approximately, you have an overnight, you come from an overnight fasting period. So because you have not eaten anything, so your ghrelin levels tend to rise. So when you wake up in the morning, ghrelin levels tend to rise slightly. And ghrelin now sends your brain a signal, and you feel that you need to eat food, so you feel hungry. So again, it's released after you sustain a prolonged fasting period. So if you haven't eaten for a while, or the longer it takes for you to eat, or the longer you fast, the higher ghrelin tends to be, because ghrelin keeps sending a signal to your brain saying, well, we need more food. And then you feel hungry and you go and you become willing to eat. But as you start eating and your stomach starts to stretch out, and you stimulate the production of CCK, which is released during a meal and acts through the vagus nerve to promote satiety. So in other words, you have innervation on the central, the nervous system. There is a nerve called vagus that innervates your stomach and also your gut, or your small intestine. So what happens is that as CCK is released, the nerve endings sense that in the stomach and triggers a signal that goes to your brain and stimulates satiety. So we have, again, two hormones that are related to, or they change on a meal-to-meal basis, and they have opposite effects. Ghrelin stimulates hunger, CCK stimulates satiety. So both of them, they alternate depending on what kind of, if you're in the fed state, you have high CCK, low ghrelin. If you're in the fasted state, you have low CCK and high levels of ghrelin. So by doing this, or by alternating the release of these hormones, your gut actually sends information to your brain. And with that, your brain can actually stimulate, either give you this feeling or the subjective feeling, or trigger that subjective feeling that you look at the food, you say, okay, I'm full, I don't want to eat anymore. As opposed to when you are not eating and you get the feeling that you're hungry and you say, well, I need food. So this signals how your brain integrates these signals and convert them into a subjective feeling. Depends on, obviously, on what state you are, and it varies on a meal-to-meal basis. So that's why we call it short-term signals, because it works from morning to noon, from noon to evening, because if you have 3 meals a day, your body has at least 3 different cycles where you have hunger, then you eat and you're satiated, then you have a period where you're not eating, then you get hungry again, and then you eat, you become satiated, and then you have another period where you don't eat, and then you get hungry again, and then you eat, and then you become satiated. So this is basically alternating different short-term signals, and I'm listing 2, probably the most important ones, ghrelin and CCK, that oscillate and alternate according to the time of the day. And your body is also capable of developing what we call circadian rhythm. So because if you adjust yourself, if you accustom yourself to eating 3 times a day, it's interesting because as you approach those specific times, you start seeing the surge for ghrelin, for instance, as you get close to breakfast time, or you have another surge close to lunchtime, and then another surge before dinner time. So you have this rhythms of release of ghrelin, and obviously this is accompanied by CCK, but CCK is dependent on whether or not your gut, your stomach fills up with food, the stretching receptors actually are triggered, and then they release CCK, and CCK through the vagus nerve sends an information to the brain. And there is another thing that we also discussed, I would say quite lengthy on Monday, which was what we called hedonic eating. Again, this CCK, ghrelin, and even leptin, these are all signals that are basically of physiologic nature. In other words, is your physiology that actually commands or controls it. But you also have some control that is based on the information you collect from the environment that you're in. And these are what we call external cues. And you can actually override all the signals. You can override the effects of short-term physiological signals that arise from the gut. So even as I said, if your gut is stretched out to the point that there's plenty of CCK and the signal is going up to your hypothalamus through the vagus nerve, and it's telling you you've had enough, you're full, you don't need more food, you don't need more food. But still you can say, well, I can still eat something, especially if it's a palatable dessert that is available to me. Why is that? Because your body's telling you that you don't need more food, but you still go on and you eat that. So that's what we call hedonic eating, and it's basically the ability that we have to override the system or the physiological system and either eat when we shouldn't or not eat when we should. So there are these are opposite mechanisms, opposite responses that you decide to do, or things that you decide to do with respect to food, when your body is telling you one thing and you're going the other direction. So you override the system. And we actually discussed about this, or we spoke about this to, I would say, lengthy last Monday, so I don't think we need much more to talk about it. I think the important message here is that you can override either long-term signals, such as the leptin signal, but also the short-term signals which arise from your gut as you eat or as you fast. And there is another important aspect that regulates your food intake that is not directly related to ghrelin and CCK as the food is in your gut, or the absence of food in your gut, or the presence of food in your gut. And this is because your body also needs to maintain glycemic control, and this affects energy, obviously energy homeostasis. Because I mentioned before, there are some cells in our body that can only operate when you have, or in the presence of glucose, because these are cells that do not have the ability to metabolize to metabolize fat. So they have to rely on glucose. And normally what we have to sustain is a glucose concentration that is within a relatively narrow range, which we could say around 5 millimoles per liter, or if you don't go by millimoles, if you go by milligrams per deciliter, is about 80 to 100 milligrams per deciliter. So that's the normal, we would say normal glycemia, because this is the maintenance of a relatively constant level of glucose in the circulation. So these organs and cells that can only survive on the base of glucose or using glucose, they are constantly, or they're supplied with that substrate. So these organs are brain to a certain extent, because brain loves glucose, but it can actually also adjust itself to use ketones. And we discussed this before. We're going to discuss a little bit more here. But your brain is an organ that really likes glucose. And if you give a regular supply of glucose, or if you provide a regular supply of glucose through the diet, then your brain will preferably use glucose. And now, if you are fasting, or if you're doing intermittent fasting, or if you're using any other strategy or dietary strategy where you restrict glucose, if you're going on a low carbohydrate diet, for instance, and you restrict the amount of glucose that comes from the diet, then your brain may be able to use, or may adjust itself to use ketones as an alternative substrate. So the brain likes glucose, but it can adjust to an alternative substrate. Now, the kidneys, for instance, there are specific areas in the kidney that it can only use glucose as a substrate, because the blood flow is limited, so the ability to perform aerobic metabolism in some specific core areas in your kidney is limited. So glucose becomes an important substrate because the kidneys cannot do well with fat. And we also have, for instance, red blood cells, or erythrocytes, which are cells that they don't even have mitochondria. So even if you have plenty of fat, it cannot use it. It cannot even adapt to use alternative substrates because it lacks mitochondria. So the main function of a red blood cell is to carry oxygen. So if it were to use the oxygen that it carries, it would defeat the purpose of that particular cell. So for that reason, I think nature took away mitochondria from red blood cells, because this way now you can make sure that red blood cells will carry oxygen, but it will not use it. So the tissues that will collect that or receive that oxygen that is being carried by red blood cells can be, or the tissues can use that oxygen that is carried by red blood cells without red blood cells using them for themselves. So that now creates a situation where, okay, well, red blood cells also need energy. So how do they actually acquire energy if they cannot use oxidative metabolism because they lack mitochondria? What is the alternative? So they have to use glucose, and they use glucose by glycolysis or anaerobic glycolysis. So red blood cells, they rely essentially on glucose. They produce lactate, and actually even under resting conditions, if you take a sample of your blood and you measure lactate, even when you're sitting there, you're not doing any exercise, you're going to see that there is approximately 1 millimole of lactate in your blood. And this is because this lactate comes mostly from red blood cells and to some extent from other organs and tissues that produce in relatively small amounts. So the point here is that you have cells in your body that rely essentially on glucose. So you have to be able to use that. So your body, again, glucose is not an essential nutrient because you don't need to eat, but your liver will produce if necessary, so it maintains a relatively constant blood glucose level. So that's why it ranges within a narrow, or it varies within a narrow range, and it's about 5 millimoles per liter, or between 80 and 100 milligrams per deciliter. So your body has to maintain. It's not only ghrelin and cholecystokinin and leptin, for instance, that regulate your food intake and energy homeostasis. The amount of glucose that is available is also an important regulator of your energy homeostasis. And again, as I said, you don't need to eat, you don't have to, it's not necessary, it's not an essential substrate, carbohydrate, but you require it for specific cells that need to survive exclusively on glucose. And for that reason, your body also triggers or affects the way how how satiated you are, and also how full you are, depending on how often you eat glucose or how much carbohydrate you have in your diet. So there are two terms that I want to make sure that you understand. One is what we often refer to postprandial period, which is the time immediately after the ingestion of a meal when blood glucose levels rise. In other words, you ate shortly after, in the period immediately after, I would say 2 to 4 hours after, 2 to 3 hours after your meal is what we call basically postprandial period. It's when blood glucose levels rise. Obviously, it will rise if you're eating foods that contain carbohydrate. But if you're eating foods that contain very little or almost no carbohydrate, so blood glucose does not even rise, it stays relatively constant. But it's still considered postprandial period. And then we have the opposite, which is called post-absorptive period. So all contents of the last meal have been absorbed from the gastrointestinal tract, but not much time has elapsed to initiate a starvation response. I think the best example of what post-absorptive period is the period just after an overnight fast and before breakfast is consumed. And this is because when you wake up in the morning, your blood glucose concentration is slightly below 5 or around 5 millimoles per liter, which means that everything that you ate the night before was cleared from the circulation. So everything, all the glucose that was in excess, if there was glucose or carbohydrate, glucose in excess was stored as glycogen, was oxidized, was transformed into fat. So everything is cleared from the circulation. Now, and you still have to maintain a certain amount of glucose in the circulation because, as I said, your red blood cells, brain, kidney, so specific tissues, they require glucose. So there is a maintenance of a normal blood glucose level. It's around 5.5 millimoles per liter. And again, the post-absorptive period is when you cleared all the contents of a meal, and now you are in that intermediary time between a meal, but you have been away from food for at least, I would say, 8 to 12 hours. That's usually the time that characterizes post-absorptive period. And the reason why your body is very specific to, and I mentioned this in terms of regulation of blood glucose levels, because you don't want to undergo hypoglycemia. So there is serious hazardous consequences that occur if glycemia falls below 3 millimoles per liter. We call hypoglycemia. Or the opposite too, if it climbs above 11, which is severe hyperglycemia, such as in diabetic patients. So your body is constantly trying to regulate glucose. So, and because glucose is directly related to the ingestion of food, especially of carbohydrates, there is a direct relationship or an important role of regulating glucose that is, I would say, cross-reacting with the regulation of food intake and energy metabolism. So if you have low blood glucose levels, your body senses it and tries to stimulate hunger, and you become hungry. I mean, you really go for food. But if it goes through, and if you maintain that low, even though the signals are sending you a message, and that you need to eat, and you still don't eat, you may actually drop blood glucose levels to a point that you pass out. So that's one of the risks of hypoglycemia. Now you have the opposite situation where you have too much glucose in the blood and it's hyperglycemia, but that's a complication associated with diabetes. More so, well, you have that's in both types of diabetes, type 1 and type 2. But it's a problem that if it's sustained through long term, it also causes severe complications. So you don't want to go into hypoglycemia and you don't want to go into hyperglycemia. So there is a range of glucose levels that we want to maintain or stay within, and this can be helped by depending on what kind of diet you have and how often and how much you eat, and also whether or not you have some other conditions that can affect your glycemia, such as I said, diabetes, obesity, or other problems that can make you develop hyperglycemia. So the average person on a Western diet, typical diets that we have here in Canada and the US, mostly in North America, is what we call Western diet. It's rich in carbohydrates, it's normally rich in processed foods, and there is quite a bit of sugar involved in the diet. So the average person on a Western diet is estimated to eat approximately 300 grams of carbohydrate. And this is considering that you divide this in 3 meals a day, and approximately 80 to 100 grams of carbohydrate per meal. So if you breakfast, you eat some carbohydrate, you're going to get the first installment of this 300 grams that I'm saying, and I'm mentioning here. It could be more, it could be less, but this is just an average, as just for us to have an estimation of what's going on. Usually people eat a lot more than 300, and I've seen people eat in a typical high Western diets, especially in North America. Usually it's even more than 300. It's about 500 grams of carbohydrate. So, but if we divide in 3 meals and we take 300 as an average, so it's about 80 to 100 grams of carbohydrate per meal. So what does that mean? So in a 24-hour period, this 300 grams of carbohydrate is enough, or provides enough glucose to replace all glucose present in the extracellular fluid by about 25 times. In other words, the amount of glucose that you have in the circulation, in your blood, in your plasma, in the interstitial fluid, so outside of the cell, that can be replaced, can be you could actually remove and put back 25 times in a day if you eat 300 grams of carbohydrate. This is just to give you an idea of how much more than necessary glucose is normally available from the diet. And obviously, you don't recycle 25 times, you don't eliminate all that glucose. I mean, you have to to come up with a mechanism that you can actually deal with that amount of glucose that is available, because obviously you don't burn out that glucose 25 times and there is no leftover. There is a large amount that remains, it's a surplus of glucose. And then where does it go? It usually goes into the adipose tissue, because the extras, well, the extra amount of glucose that is not used by the cells, that is not stored as glycogen, it goes into the adipose tissue. So because you cannot completely exhaust or utilize all the glucose that is available if you take in, like in this example, 300 grams of carbohydrate, which is easy. People eat even more than that without much difficulty. So this is I would say a very good example, a very good estimate of what normally has and how much your body has actually to how much glucose is available and how your body has to deal with it. And again, because you don't burn all this glucose, because otherwise you would be in a very high metabolic rate. Usually you have a surplus of glucose that is not stored as glycogen, is not oxidized, so then your liver converts into fat, and then it goes in the adipose tissue. So what are the organs then and I think we have to some extent discussed this when we spoke about carbohydrate metabolism. So we have basically 3 organs that really play an important role in regulating this energy, metabolism, energy homeostasis. And it's after a meal. So we have the insulin stimulated glucose disposal. So where does the glucose go after you eat? All this glucose that is available, as I said, it would be in theory sufficient to replace all the glucose 25 times a day. So obviously you don't oxidize, you don't eliminate that glucose. You have to be able to manage it somehow, because there is obviously a surplus. So liver stores some of it as glycogen and also oxidizes some of it. So, but a large portion of it is stored as glycogen, which is important because this is what keeps your liver is the organ that really maintains your blood glucose levels constant throughout the day, no matter what you do. If you don't eat carbohydrate, you're in a very low carbohydrate diet, your liver keeps sending glucose out to maintain blood glucose levels. So liver is an important organ, partially because it promotes glycogen synthesis when you have a surplus of glucose and converts it into glycogen. So it's stored there. So, but there is also a large portion of glucose that goes into the muscle. So the muscle also takes up that glucose, and provided that you're in the resting conditions, it will also store glycogen. So these two organs, but they have limited capacity to store glucose, because for every 1 gram of glucose that you store, you also store 3 to 4 grams of water. So there is a limited amount that can go into these organs, because otherwise it would expand so much the organ that it would cause problems to the organ. For imagine if you put 200 grams of carbohydrates, of glucose, of glycogen in your liver, you have just as water, you're going to have approximately 3 to 4 times that. So that 200 grams of glucose or glycogen would be added to another 3 times. That would be 600, approximately 600 grams, or it's about 4, could be 600 to 800 grams of water. So you almost have a kilo added to your liver just by combining the glycogen with water. And the same thing is in the skeletal muscle. So there is only so much you can store as glycogen, because otherwise it causes a major issue and the organ is not viable anymore. So liver and muscle are two important organs that store glycogen, but they have limited capacity. Muscle is important because we have a large amount of muscle. It's about, remember, for men 40%, for women 30% of body weight. So you have a large compartment to store glycogen. But still, every muscle stores a relatively limited amount because of this ratio with water that usually comes when these organs store glycogen. And then we have the adipose tissue. So the adipose tissue stores limited amounts of glycogen. What the adipose tissue really does is store fat. So what happens is that additional amount of glucose goes into fat, and then you take, for instance, the adipose tissue is very good at taking glucose, converting into glycerol. And remember, glycerol is the backbone that your cell uses to sterify 3 fatty acids and store as triacylglycerol. So the adipose tissue is rich, is professional, I would say, in storing fat in the form of triacylglycerol. And it does so because it takes glucose, makes glycerol. Now the fat that also comes, that is available in the circulation, and if you eat fat from the diet, so now you have fatty acids that can be used to sterify to that backbone of glycerol. So the adipose tissue, the combination of the adipose tissue storing triglycerides, the skeletal muscle storing glycogen, and muscle and liver storing glycogen, you pretty much take care, or you manage the influx, or the amount of carbohydrates that comes in, and also of fat that comes in from the diet to these organs via circulation. So these 3 are important for, again, glucose disposal, and they respond, all these 3 organs depend heavily on insulin. So when you eat a diet, when you eat a large amount of carbohydrate, insulin goes up, tells the liver to synthesize glycogen. Skeletal muscle, the same, and tells the adipose tissue to take up glucose and use it to sterify fatty acids and form triacylglycerol. So these are the 3 main organs that take care, I would say, of the additional surplus that is not used by any of the other organs that I mentioned before. But you can also, it's also necessary to maintain a relatively constant blood glucose level around 5.5 millimoles per liter, 80 to 100 milligrams per deciliter. So those cells, such as red blood cells, your brain, kidney, specific areas of the kidney where only glucose can be used to produce energy. So again, these are important because regulation of food intake and whole body energy homeostasis is a very complex process in which you have those hormones that play a role in the short term on a meal-to-meal basis, and you also have the long term, which means that how big or small your adipose tissue is. And obviously, to be able to manage this, because you have the signals that tell the brain one thing, you have you feel hungry or feel satiated, but the organs that actually manage the substrate that is available for energy is basically liver, skeletal muscle, and adipose tissue, because these are the organs that play the most important role in managing all this information, and not only that, responding to insulin depending on how much carbohydrate is available on the diet. So it's a much more complex system than we usually think, and it depends on the signals that arise from the gut on a meal-to-meal basis, but also depend on the regulation of blood glucose levels and the size of your adipose tissue. So with that, we are going to stop here today, and we'll continue. I think we are going to have 2 more lectures, one on Friday and then another one on Monday, which is April the 12th. So in 2 more lectures, we will pretty much complete the material for the course this year. So thanks for watching, and I'll see you, or I'll talk to you again on Friday. Bye now.